Okay, hi everyone. We'd like to start. I hope most of you are not tired after so many talks we had in the last few days. But nevertheless, we're very happy that we have Michael along with us today. He's visiting Israel, so we took the chance to invite him, although we had just finished a long conference. So Michael is Thomas and Susan Murphy Professor of Neuroscience at NYU School of Medicine. He completed his graduate studies with Barry Connors at Brown University, where he investigated the role of electrical sciences in mammalian brain. During his postdoc, he worked with Michael Fee at MIT, and then where he began to study songbird, assisting to uncover the several network properties that give rise to learned vocal representation. Basically, he's interested in circuit underlying skilled movement, often in the service of vocal interaction, and he studies songbirds, human vocalization, and also human speech and mouse vocalization. And one of his celebrated work was finding the singing mice and basically how they make conversation with each other. So we're happy to have you today. Thank you for coming.
Thank you. I appreciate everyone, after sitting in a room for 2 days now? 4 days. How are you all awake? Feel free to get coffee or take a nap. It's all right. I'm very excited to. I was giving a talk at Tel Aviv 4 days ago, and then I gave a talk at Weizmann 2 days ago, and I'm here, and I thought, I have only visited this building when it was about half done, which I think was a period that spanned about a decade, right? But it's such an amazing building, an amazing science so far. I've loved all of the interactions. So thank you all for coming. It's about 6 times more people showing up than I had anticipated. I have 2 talks queued up. One is songbird and kind of broad lessons that we can learn about local processing versus long-range inputs. There's another one about humans and singing mice. I kind of threatened to give the second one, but I think I'll stick with this because in some sense, a lot of the inspiration that I've gotten are from people right here in Israel that have thought about large networks and behavior and what they're doing. Remove it from me. You need that? Or? No. Amazing. Okay. So I think I'll give this talk, but if anyone's interested in some of the human stuff, I'm happy to discuss that later, and singing mice and all of that. Everything's moving forward. Too many animals, too little time. Okay, so the big question that my lab has been trying to wrestle with for years now is how does the brain give rise to skilled movement? So this is decidedly my brain, but not me playing tennis. And so how do we take something like a tennis swing or a musical piece and encode this movement sequence, skilled movement sequence in the brain? And we luckily have a creature that does this really well, along with, hey there. And we know something about the brains of these animals. And so this is the musical piece that this animal is an expert in. We were very excited about the song learning process. But for now, what I'm going to show you is a movie of an adult zebra finch, which is this guy right here. This is a male, that's a female. The male will perform his courtship song, and hopefully things will unfold here. This is the sonogram, so this is frequency, high notes, low notes, and this unfolds in time like that. Fine, so if you haven't woken up with that, then I can't help you. Okay. And you can see that this is a kind of sequence of sequences. There are different syllables that are performed as part of this courtship song. I've now decorated each unique syllable with a color, and we can get a very close look at these syllables, as you see here, and see that the structure of these, really at a kind of millisecond by millisecond level, in this bird that spent hundreds of thousands of trials learning his song and performing this, then for a female, is really quite precise. Right, so we have this behavior, it's a tractable, really precise behavior. And there have been 2 papers that have been really inspirational to me over the last 50 years. Many papers have been inspirational, but these are the 2 hallmarks that have really set my own research trajectory, at least with respect to these songbirds. And the first one was written by Fernando Nottebohm and colleagues back when I was 1 year old. And what Fernando did is he saw a group of brain regions here that are all necessary for producing the song, right? So not only is it a very tractable behavior, it's actually produced by this tractable series of regions. So it's great that human neocortex and neocortex in other animals can be multimodal, but what is great is that these are doing that behavior. So we have it kind of cornered, if you like. And the major dynamics that I'm interested in can be shown in this beautiful, now historic paper from Michael Fee that he wrote many years ago. And what Michael did is he stuck metal electrodes into the songbird HPC, this premotor area, and he could record one cell at a time, back in the Stone Ages of recordings, right? And for example, this green cell is active at just this one moment during the song. Okay? And as he's recording, and the animal is singing over and over again, you can align the behavior of that cell to the song, and you see the actual variability of that burst is very low, really sub-millisecond. And record, in this case, a group of 8 cells, and relate this back to behavior and see that this forms a sequence as you've reconstructed it in that way. So now in the era of large-scale recordings, and this is also somewhat quaint, this isn't quite neuropixels 9.6 or whatever it is now, this is 126 channel probe that we put into HPC. We can see 20 neurons, all recorded at once. These are fairly sparse cells, difficult to record, but these are all projecting to this downstream region that we care about those cells. And then that allows us to look at a trial-by-trial basis, and we can see that there's always the same set order, same set timing, even across these trials. And so now what we want to do is start to ask the bigger question. This is mainly the thesis work of a very talented student who's going to be starting in Gilles Laurent's lab next month. Her name is Margot Olamali. And what Margot wanted to know is, if we have a series of these bursts, are those bursts and their timing and their relative kind of network dynamics writ large, is this something that is the product of local interactions within the circuit, or is this something that is really generated by a larger scale network to create those bursts? And let me lay out a few different hypotheses in exactly what I'm talking about. So here are some of the other players. This guy will be the star of the show later in the talk. This is the brainstem. These guys will go down to respiratory musculature for breathing, as well as the musculature that changed the tension on the vocal apparatus. This is the primary cortical motor region, and then this is the premotor region, HPC. So you can imagine this extreme model that was proposed, part of a neuron paper a few years back, that it takes a village to create every single burst. So the burst, it travels around this polysynaptic loop. It's another burst, travels around this polysynaptic loop. Here's another burst, et cetera, et cetera. This is a model that I favor, or at least favoring, not to give too much away, that there is some kind of local interaction that's important for generating the kind of sequences that are at syllable scale. But then for us to go from one syllable to the next, we may need this kind of long kind of polysynaptic loop. And of course, the extreme model there is that everything, we're one big brain in a jar, but there's lateral interactions like this that can form a sequence within this area. So I think whether you care about songbirds or not, the idea of local versus long-range processing is something that we really have to wrestle with as a community, and how can we really tease these things apart. So as I mentioned before from Fernando Nottebohm's work, if you lesion any of these, you hold a pin out from any one of these structures, the song falls apart. And I can play a little bit of what that sounds like. This is what the standard song sounds like. And this is what the lesion sound. You're welcome. Okay, so the problem is we pull this pin out, and now we can't really study the system, okay? Because the behavior falls apart, and now the whole thing is no longer able to be investigated. So how can we disentangle this in this kind of over-interconnected case? Well, we're not alone in this. So Adam Hantman, Carl Svoboda, others have fought with the same problem, that there's motor elements, that areas like gambling that are important for skilled movements, either hand movements or licking movements. perturb one, it really affects the other. I call this cortical thalamic codependency. Right? So these things are all really tied, and it's hard to know where one begins and the other one ends. So this is a quote from Hammond. Motor cortex activity patterns during movement are crucially molded by its embedding within a vast and distributed network. So we're throwing up our hands and saying it's complicated. So what we decided to do is take one step back in order to try and take 2 steps forward. And we wanted to have a method for opening up the loop to really characterize this in a behavior that's not going to fall apart. And so we decided to think about sleep dynamics. Okay? So sleep is something that has been studied by a lot of labs, and really beautifully so. And so this is work from my good friend Albert Lee, when he worked with Matt Wilson. And what Albert found is that if this animal's running down a linear track, and this is the activity of some CA1 neurons, you can see that they're bursting at specific moments along that track. And when this animal goes to sleep, there's in this case a hyper sped up, a 10x sped up, a little snippet of a replay of those movements. So Dan Margoliouth saw something that was kind of similar to that. So here he's recording not from the sparse sequential activity, but that kind of primary motor area that's just one synapse down, called RA. And that bursts about a dozen times during the song. You can see this burst pattern. And what Dan did is he turned off the lights at the end of the day, he was leaving, and he started hearing a bunch of bursting activity, and he found, oh my God, this thing seems to line up pretty well with what's going on during singing. So we wanted to study that. We wanted to study that nighttime activity and do some hypothesis testing on this nighttime activity in order to get real answers. Because before we pull out the thalamus, the song goes away, but maybe this is going to be more resilient than that. We can actually use it to get it network structure. And so, returning into our silicon probes, HBC have these sparse, difficult to record from cells. Here's 11 of them. You can see there's activity that happens during the night. I can zoom in on that activity. It looks a lot like Albert Lee's activity. What we don't see is any kind of speeding up. So we don't see this kind of 10x speed up, it's kind of 1x. And I'll put a fine point on that in the next few slides. If we look down here in RA, this is a very dense code. It looks like a QR code or something like this, like you're getting into the concert by showing RA activity or something. And so what's good is that each one of these burst profiles represents about a dozen presynaptic HTC neurons. So it's a way of cheating the system, if you like. Right? We can get 12 neurons for free with every neuron here. They're big, easy to record from cells. So we get a great handle on the song system by just sticking our electrode down here in RA. There's bursting activity. There's also single spikes. These guys happen in slices in synaptic blockers. So pay no attention to the single spikes. It's just the property of these cells. But the bursts, I think, are really interesting, and I'll show you why I think that is. So first of all, I do want to make the point, with this arms race, Moore's law of electrode count, how many is enough? Right? Well, we can actually do a back of the envelope calculation here to say how many do we need to answer this question, and it's a comforting thing. We don't say let's get 10,000 neurons and then figure out what our question is next. We can say we need 10 millisecond accuracy to really distinguish between our models. Okay, fine. And how many cells do we need to really get that kind of accuracy? The answer is maybe 2,000 or so. Great. Because each one, again, is encoding the activity of a population of those sparse sequential neurons upstream. Fine. Okay, so we do a very simple decoding algorithm here. So we can take all the activity happening during song and sleep. We ignore the single spikes. We just go burst or no burst. And then we say, for a given moment in sleep, for this little 5 millisecond window, what part of song does this most resemble? And we can just run that series of ones and zeros throughout song like this, and we say, well, it most resembles this part. And that's fine, and you can say, well, maybe you got that, give you a chance, maybe we did. But we look at the next moment during sleep, and we can see it actually resembles really well the next moment in song, and the next moment in sleep resembles the next moment in song, and further like this. And so what we see are these moments in song that seem to be little snippets of dream that can read out at a very high temporal resolution. Okay, so we have these moments of sleep replay activity, and this is just another example of that. We see this activity, and we have these moments during the night. like this, that the bird is dreaming. We wanted to ask the question then, where is this activity coming from? Right, so if we send a test signal in here, and then we can, for example, knock out thalamus and find out what comes back. I'll tell you why we think that's an exciting thing to do. But first we wanted to find out where is this activity born. And so we used an old trick that I played with many years ago, and that is focal cooling. You can actually change the temperature of this part of the brain, and it's a quite selective manipulation. You don't change these other areas, they're far away. It's something about the way that the bird brain is made, that these are not only really critical areas for the behavior, but they're scattered across the brain, so we can really treat them one by one. And if we cool this area, and that's really the source of this nighttime activity, we expect the slopes of those reconstructed dreamlets to get slower, to be less when we cool down that area. They're generating this kind of nighttime pieces of song replay. Of course, if it's elsewhere, we expect to see no change. And what we find is exactly something like this. Even though we're reporting from RA, which is one synapse down, this kind of dense area, we see by cooling HVC, in this case we've cooled it by about 8 degrees, we see the slope of this dream get decidedly more shallow. And we can see this monotonic relationship looks like this, depending on the amount of cooling we do within HVC. So I think we've got the sleep dynamics cornered in terms of where they're being born. And for those who remember this paper where we can change the behavior itself, like by altering the temperature of HVC, we see exactly the same change per degree, about 3% per degree. Okay, so sleep activity starts there. How does it progress? Let's knock out thalamus, and we have certain ideas of what we might predict if any of these models are correct. Okay, if things are a distributed model, then we expect to start the party here. We could knock out thalamus, and we should stop these long sleep dreamlets, right? We should abolish sleep replay. Okay, so let's look first at that prediction. What we've done is we put a stimulating electrode within the thalamus. We let the animal sleep throughout the night, 4 or 5 hours. Then we do an electrolytic lesion in thalamus. So the electrode is there. We burn out the thalamus, and we ask what happens for the rest of the evening. And what we find, identifiable replay sleep events keep on trucking. And then the bird wakes up, and he can't sing. So this is impervious to that manipulation, but clearly the behavior is compromised. We can also do histological verification that we've burned out all of thalamus on that site. So these two are a bit nuanced, but I think I want to be really clear about the predictions for these two models. We can reconstruct these small sleep snippets, and if this long kind of feedback pathway is needed, by killing one aspect of this pathway, we would expect the sleep snippets to stay within their fence. Right? We'd expect them to stay within this kind of syllable. Right? And this continuous model, if there's enough information here to make it through the entire sequence, then by killing thalamus, they should continue to cross those boundaries with impunity, as we see during natural sleep. What do we find? We find something that is really consistent with this continuous model. So the snippet starts here with this kind of filled circle. It goes into the open circle. These are a bunch of little sleep snippets that occur under normal conditions. We burn the thalamus. It's a tough thing to have that happening in the middle of the night. But and what do we see? We see it continues to cross these boundaries. Okay? So each dot here represents a single syllable boundary for different animals shown in different colors here. And you can see the pre and post lesion probability of crossing those boundaries is best described by this unity line. It supports a continuous model. Okay, so the idea here is that we have propagation occurring locally within the structure throughout the duration of the song. This tells us that there's sufficient information within this local HPC network to propagate sequences. And the moral of the story for us, what we were excited about here, is that looking outside of the context of behavior enables a clear dissection of this circuit structure. I want to give a little tiny vignette after this and then maybe pause for questions before we get to the next section of the talk. So I want to tell you something kind of general about the songbird brain. This is something that Nikki and I were talking about earlier today. What about connections across hemispheres? These guys, at least at the level of the forebrain, lack them completely. So HVC stands alone, RA stands alone, and these are here almost like a natural split brain patient. This is an actual drawing of a human split brain where the corpus callosum was cut. And so this is a quote from one of those Sperry papers back in the day. I wish it would put my right for the thing I wanted, but the left would come in and then kind of fight, almost like repelling magnets. So there's this amazing literature of what happens when these two brains are acting in very different ways. So what about the songbird? Well, we know from just lesion studies, if you lesion either the left or the right, the bird has major problems with his song. But we really don't know. Like, the problems seem to be incrementally more major if you lesion the right. So we always put all of our electrodes here. We've really ignored this side. This is the dark side of the moon. We've never really recorded there. Let's look at both sides and find out if we can learn anything by recording left and right. So here, recording from that QR code area in RA, there's a bunch of bursting. We can use some analysis that I don't want to spend a lot of time on. This is just to say that if we predict the activity on the left by looking on the right or vice versa, we do a very good job and actually a sub-millisecond correct prediction. And so this tells us that these things are really going in lockstep, left and right, during the performance of song. But this goes out the window for sleep. So you can see that this, again, you can ignore the single spikes, both on the left and the right. There's bursting here that's not happening there. There's bursting then on the left that's not happening on the right, as you can see. Right, so it seems like there is replay happening on one hemisphere but not the other. And this is very different than unihemispheric sleep. So when we look at other measures of arousal, they all seem to be going up and down together. But if we look at the statistics of this at a more kind of tens of millisecond level, there are some discrepancies. So then we can decode this, right? We use a template matching method to say what is actually in those bursts. How does this relate to song? And there are moments like these long replay events here where they seem to be replaying different, the exact same part of the song. You can see that with the overlap of left and right. But there are many other moments where left and right are doing decidedly different things. So the red tells us where in this song, we should have the sonogram of the song here, where this right-hand side is being replayed, and concurrently with a very different part of the left-hand side song. There are also times when the bird is clearing his throat or making other types of vocalizations. We can decode those too, and those are often happening while the bird is replaying his courtship song. So it seems like these two sides of the brain, for the most part, with some exceptions, are acting as very different brains during the night. And so if we look at cases where there is bursting happening that seems to be replaying the courtship song, we can ask the question, how close are these in the courtship song? What is this distance between what the left is doing and what the right is doing? And this is over the entire night of sleep. So things kind of have a tendency to stay around zero, right? But they're often wildly divergent from that. And so we're excited to see times when there are structures and aren't structured. Maybe thalamus is kind of stuttering on or stuttering off. Thalamus broadly is pretty disengaged during the night. And we'd like to follow up on this a little bit. But I wanted to just say these two brains are acting as very different agents during this time. So this is kind of new data and also a summary. So this is looking at this with a cross-parallelogram, saying if something's happening here in the song on the right, what's happening on the left, and you can see there's a ghost of the central line telling us there is some kind of vague coordination, but really it's anybody's guess. You know, this kind of replay can start and end anywhere, and it can do so quite independently across these two hemispheres. Of course, if we do the same analysis during singing, things are beautifully on this middle line, precisely coordinated. So to try and solve this mystery, and to try and, I think, bring these two first kind of chapters together, we wanted to look directly at thalamocortical interactions during singing. But I want to pause here and find out if there are any questions. Yes?
So the activity in U, you see fast gamma?
We do see gamma-like activity in UVA. So that's not our work. That's work from Michael Fee's lab. But I'm going to put a very fine point on UVA in exactly the next part of this talk. So come back to me with exactly that question later. Great question. Yeah?
How does the heterogeneity evolve along the night, and what happens if you sleep deprive the bird? Will it be less coordinated?
It's a good question. Haven't done it. What I'm particularly interested in, so this is almost using sleep, not almost exactly using sleep as a tool, but I think there are lessons there about how sleep is used in the young bird as it's learning. So these are, it's an asymptotic case. This is an adult bird that has learned his song, that's probably maybe using this to maintain its song, maintain a high level of expertise. Ofer Chernikovsky did some beautiful behavioral work many years ago, where he just looked throughout the day at how close a young bird's song was to the tutor, typically the father. It gets better and better and better throughout the day. Then they sleep, it's bad. And then it gets better and better throughout the next day. In fact, superseding where it was the day before. And then they sleep and it goes down like this. And what he found is the extent to which things go down during sleep correlates with how well that bird ends up doing in the end, right? So no pain, no gain in some sense, right? And he referred to this as an annealing process of things heating up, cooling down, and actually strengthening the behavior at the end. So we're doing recordings in the young bird, and we're very excited to know about how this kind of rehearsal, which seems to be taking a half step back, could be useful for the actual broad learning process, especially considering, the bird isn't hearing himself during this time. Importantly, the bird does flex its song muscles during this time. It doesn't change its breathing at all, so one thinks that if one could apply pattern breathing into the air, from the air sac, you'd probably be able to elicit sleep songs that would sound really crazy because both sides are doing very different things. Any other questions? Stretching, no question. Okay, good.
What about coordination during flight? If they need like the two sides to coordinate?
Yeah, I agree. And I'll admit that the flying system is something I don't know whether there is a direct communication there. But what I can tell you, and what I think is important for thalamus is called UVA. These names are terrible. We need other names. But UVA was first described because the nucleus looks like a grape. It's 750 neurons, very small, very deep, hard to record from. And but UVA is given bilateral information, so that's one place where things do cross. So if thalamus is coming up and telling cortex something, it's an informed piece of advice because it knows what the other hemisphere is doing. So even though there isn't a direct line that says I'm going to hardwire these two things to act as one, UVA is something that you want to listen to. Now I'm going to wonder why my computer has. Let's see. All right. Why don't we restart the program? Okay. So okay. So let's perturb things, shall we? This is, we realized that thalamus was interesting. I don't need to convince many of you that thalamus is interesting. But this is a very simple trick that we did, and that is we administered a pulse of electrical stimulation at different times during the song. We did this in cortex, so in this premotor area HBC. We also did this in thalamus. And wherever we administer stimulation, you can see these golden vertical lines. The song broke. Okay. What is stimulation doing? I thought we were going to talk about opto or something. Okay. Well, what's nice is at least when we measure this locally with the silicon probe, we find that excitatory cells, and these are basal ganglia projecting excitatory cells that have a non-zero baseline firing rate. We can look at their activity. You see that these things are popping right along, and then about 200 milliseconds. So you basically paused these excitatory neurons following the stem. The stem is at time 0. And why? Because when we're recording from local inhibitory interneurons, we see that stem does a very good job of activating these, often with really high frequency bursts like this, that can even occur at somewhat different times throughout this 200 millisecond period. So we're demystifying what I think local stem is doing. When we talk about how stem may be shutting down specific motor areas, this is how, because these interneurons are really excited by the stem, and they do that. And so let's go back to our stem manipulation and take a somewhat closer look, because when we take a closer look, structure is revealed. The stem times are shown with these golden ticks like this, and like this for thalamus, and we can see where the song is breaking. When we look in to cortex, we see that any time along that continuous chain of synaptic activity, right, we can break the song. And in fact, the delay is about 40 milliseconds, which makes some sense. That's enough time to marshal local inhibition, and there's going to be a motor delay of about 20 milliseconds. So as soon as HPC shuts down, it's going to be 20 milliseconds for the muscles to get them out. What about if we stimulate thalamus? So when we do that, we see a different pattern. Right? We see that the song breaks, but it breaks at these very specific moments, these very specific breakpoints. And the breakpoints correspond to the end of syllables, or sometimes at transition points, like in this purple syllable at the very end. It looks like two conjoined portions of the syllable. You can see there's a breakpoint right in the middle, functioning here. And the way we interpret that is if we silence thalamus temporarily, HPC can act alone. It can keep rattling on until it's time for thalamus or something to kick on that next syllable. Okay, so this thalamic disruption suggests a role of thalamus for initiating different parts of this sequence. So we can't pick out gamma with this measurement, but there's some beautiful pictures, so we'll get that. This is one of those HPC neurons that's sparsely bursting. Thalamic inputs are these big, giant, beautiful synapses, I should say micro, not question. And these big synapses are coming in and they're active invariantly during song, as well as some other vocalizations. But what we were really interested in is to find out who's listening, who cares about the thalamic input. So what we don't have is like a layer 4. So if you imagine barrel cortex, there's going to be a beautiful layer 4 that's getting a lot of thalamic input. Of course, different layers get different types of thalamic input. But this is really all mixed up, salt and pepper. So we had to do our own homework and say, here are some neurons, we can stimulate thalamus, and we see this one's lighting up. These others are not. We found the needle in the haystack. Okay, we call this a putative thalamorecipient neuron. We can see that when we stimulate, for example, this cell, we get this really nice response. It looks like this, versus, for example, this other cell of the same type, where things are quite flat. The thalamorecipient cells are about 15% of our population, these thalamically driven guys. And now we have to ask the question, are these thalamically driven neurons doing anything special in the network?
So,
Yeah.
So, I mean, you activated in the you're stimulating the thalamus, and this is the HPC recording.
Yes.
Why can you see any sign of inhibition? I mean, in the response. Normally, you would expect some.
Yeah. So when we did the inhibition thing before, that was local HPC stim and recording, and we stemmed a lot. We basically gave a big titanic like this, right? And things were shutting off. Here we didn't do that.
So you think there is no, I mean, normally in the cortical system there is a feed-forward inhibition that is very strong and that can dominate this excitation. You don't think in the HPC?
So we look very carefully here, and so there are about 9 different inhibitory neurons that we believe transcriptomics and I'm beginning to. And we're trying to understand what different types of interneurons are doing. We know that PV cells, like in other areas, are maintaining this strong feed-forward inhibition. So indeed, that does exist. And yet, we still find this. So these cells, we're very excited to know, I call them kind of functional thalamorecipient cells. right? I'm not saying that they're getting a giant axon and the rest of them are not, but maybe these are the ones that inhibition is allowing to fire for whatever reason. There are a few, these could be very excitable cells, right? But let's tell a story about those cells. So we want to find out if these thalamo-recipient, putative thalamo-recipient cells are doing anything special in the network. So what we could do is do 2-photon imaging while this bird is singing. You can see this bird is singing to his partner. We can basically teach these guys to do this under the 2-photon microscope. And this is what we see in a bird that's singing under the microscope. We see these are 5 neurons. These 3 are the premotor, very sparse guys that are firing just once during the song, like this purple guy, boom, boom, boom. You can see it's always firing at the same time. These are more complex. These are going to the basal ganglia. They may fire more than once during the song. And what's great about the songbird is that we can break all the rules about how to image, right? Because calcium imaging is supposed to be very slow. Well, the beauty is we don't really care because we can look across trials and get a better and better estimate for when different spikes might occur. Because this thing is an obsessive compulsive animal. Singing this thing over and over again, the spikes are happening, we know this from electrophysiology, all at the same time. So let's do a signal detection thing and let's just record a bunch. So these are times when our laser happens to be hitting that cell there, and that's shown on the sonogram with these vertical lines, and we have this kind of estimate of what we see in terms of a response. If we look again, we see a different estimate, and we can just look across these trials until we have, in this case, a bunch of trials, 23 trials, and we see something emerges here. There's an event that's starting around here, here, and there. And we worked with Liam Paninsky. He uses an MCAC approach to say those are happening right around here. Good. So we can estimate when these cells are active in the bird and ask if the thalamo-recipient ones are we can call thalamically driven cells are active at any specific special time. This is an example of a thalamically driven cell. We put our stim away at this point. We've already characterize that cell as thalamically driven, stimulator off. This is just what's happening while that bird is making the song. And this is an example of a cell that's not driven by thalamus. These are 86 of the premotor cells, and when they're active throughout the song. And this is just our estimate for where we think the onset of that activity is. So it's a somewhat worse estimate than dash that is shorter, but we do really well because we're looking over, in many cases, dozens or 50 or more trials in some cases. So what do we find? We find that these thalamically driven cells seem to be active at this period here. Okay? And that period is with respect to the onset of the syllable. The onset of the syllable is at 0 milliseconds. Right? And so this makes a lot of sense. This is what you'd expect to be roughly motor delay. And it seems to be initiating or potentially active at a time we would expect to initiate that syllable. We also see a group of these thalamically driven cells that are active at the end of syllables and may be involved in actively terminating syllables as well. And so the way that we think about this is that thalamic input engages cortical circuitry in the songbird by targeting these starter neurons, okay? And they're active at syllable transitions and facilitating this bilateral synchronization. The thalamus comes in, there may or may not be an intermediary there, that then kicks off these starter cells that then can go throughout the syllable and maybe even actively terminate it. This enables behavioral chunks from this otherwise continuous neuronal substrate. So I've shown you during the night, there are these dreams that can cross boundaries, even if we've taken off thalamus. So we think that there's enough information to get throughout the entire song. So almost like a big strand of DNA. And this is just encoding one gene or another, if you like. And this is consistent with the role of motor thalamus as an initiator of sequential behavior. And this is something that has been predicted in some beautiful theoretical models here by Sean Scola's group, and Carl Svoboda has talked about ghost signals, and thalamus plays an important role in that as well. So we think of this, we use a vinyl record analogy, that there's information at the level of HTC. The thalamus is involved in actually putting this needle on the record to play that specific song. Are there any questions about this part?
Yes, what takes the end of the sequence?
The end of the sequence? So the end of the kind of the very last syllable of a specific batch?
How does the circuit know when to stop?
Yeah. The answer is I don't know. So what I can tell you is that, so we have this kind of intuition that some of these thalamically driven cells may be involved in actively stopping a syllable. And in fact, we do see some of those thalamically driven cells that are active at the very end, the very last syllable when the bird sings. So typically the bird will have, let's call it 4 syllables, A, B, C, D, and we'll approach a female and go A, B, C, D, A, B, C, D, A, B, C, D, A, B, C, D, and then it's done. And there are some cells that are like these thalamically driven cells that we see when we activate thalamus that are at the very end of all of that. So it could well be that these do play a role in actively shutting things off. Are you guys texting each other?
No, you said, yeah.
Oh, okay. Oh, yeah. You're allowed to step around. You don't have to turn on. Yes.
We have a related question. So, if you can repeat the same song thousands of times over, would you be able to use voltage indicators instead of pressure indicators to see the exact spike time?
What other information will voltage indicators give us?
You'll have the exact spike times, right? You could tell the spikes.
I think of this, we know a lot about the underlying syntax, neural syntax, which is there's really no firing at all. Then there's a burst that comes in at 600 hertz and then back to nothing at all, right? So in the end, spikes within a burst, as long as we know when the burst is happening. And this does a very good job of that. If we look at the middle of that burst, or even the first spike of that burst, they're jittering by less than a millisecond. So although voltage indicators may help us get published in Neuron or something, they aren't going to help us with our question. I don't want to be too dismissive. I think they're fantastic for a million other questions. And I talked to Mickey about this earlier. I'd love to know about dendritic computation in these cells. They're getting pounded by synaptic inputs all the time, and yet they're giving a very specific, well-timed output. It would be amazing to do that in the dendrites, absolutely. Yes?
So just to figure out the
Sure.
So the experiment during sleep, you do these sequences in HVC, which now you think are all actually representing one syllable. And then the jump between syllables is consistent with the loop through the
So I think there is a substrate. Train tracks, use whatever metaphor you like, that is exposed during sleep. It says, for all of the tricks that this bird does, it is there, it is part of one continuous neuronal thing, right, in that wire. I don't want to say sin fire chain, but it is in there and it's resonant to that area. Thalamus is able to hop onto that substrate and start and end things, almost like a train going from one station to the next, right? So I think it's different than saying there is enough wiring there to get through this one aspect of the song, and then enough wiring to get through another aspect of the song, and they exist as distinct microcircuits. And thalamus is able to trigger this microcircuit or that microcircuit. It's actually jumping onto a large, grand structure, right? We're working with Winfrey Denk now to try and see what that structure is, right, on a large scale. And thalamus can actually key into different aspects of this structure and hop on and then run this structure until the end of this specific part of the behavior and then hop off, right, like a passenger on a train. I don't know, I've given 3 different metaphors. I'm sure there's some kind of interference of, yes?
In the model of, I cannot pronounce the name, is Scholar, Shona Scholar.
Oh, yeah.
The first author I cannot.
Yeah,
that's right.
Yeah.
So, as far as I remember, there was a role of, go back to inhibition in this, the transition of syllable.
Yeah.
How do you?
I think there has to be inhibition. I think, so right. I think there's this, which I think is the backbone of what's going on. I absolutely think inhibition is involved. I think it's critical. And I'd love to talk to you more about this, especially to talk to you more about this, because you've done a lot of beautiful work in this. Yeah. So
Two questions, but I don't know if you want to finish or.
I have just a dessert at the very end. Not that you guys need any more food, but
Okay, so what you showed us before that RA, you quite did not influence. So are you suggesting that the control itself, like what goes after what, arranging the syllabus, or another area?
I think it's thalamus. So one thing that I'd like to do. So the problem with this bird is that it often just has the syntax of ABCD, ABCD, ABCD. There's another very, very related bird called a Bengalese finch that's kind of jazz, right? So that bird can go up and say A, B, B, A, C, B, B, A, C, C, and it's a kind of probabilistic transition between this one and this one. And what I think is probably going on is there's the exact same substrate underlying this behavior. Like the entire complements of every aspect of the behavior is there within this area. And the thalamus is able to jump on that substrate, and you just have a more creative thalamus, more restless thalamus that can actually make variability. And it'd be interesting too for more complex behaviors if you can initiate multiple behavioral programs, right, to achieve a kind of, RGB, kind of any kind of behavior. This is just one. But I can tell you, and in talking to Takaki Tomiyama, who's a good friend who thinks about skilled movements, including skilled kind of movements of a joystick, he can image thousands of cells. He gets a dozen or so, 2 dozen, that are active right at the beginning of the push, okay? And he can activate those, and that makes the animal push. So there's, you use some kind of 2-photon tricks to only get that one population. He tries to get other populations, they don't do this. And what he finds is the thalamus wires in preferentially to that starter group of cells, right? So in fact, he was mad when his paper came out. He said, "You scooped me," and I didn't. But the idea of thalamus actually really being synaptically positioned to engage different aspects of behavior within this motor structure. That's the general point.
Okay, and another question that relates to the Jaco paper.
Yeah.
So the idea there is that every time you start a new syllabus, the thalamus puts the input to put the autonomous part of the system into the correct initial conditions.
Right.
So unless those initial conditions are really close to the final stage of the network, either that there is some time, there should be a pulse, because it was pretty long, or the thalamic input is very, very strong. It's order to drive there fast in the neural dynamics. Is there any evidence of any of this?
Something I need to think about to know what initial conditions are. I mean, there's certainly moments in this bird's life where he's seeing the female, he's flush with kind of neuromodulators. Things, cells depolarize. They go into a very specific state. But we should sit and talk about how many of those things can be mapped onto this. And also, it's a model. I feel like, all models are wrong, some are useful, right? This was extremely useful and actually pretty darn
the
right.
idea that you want to drive APC to a certain point or make it react to either have to push it really strong or turn on the external input and wait to start this.
Yeah, it's a great question, and we should talk about that. So, final dessert. So we work on a lot of animals. I didn't talk today about the singing mouse and human. Talked a lot about this guy. But we're really trying to push on the budgie. So the budgie is a parrot. It can learn hundreds of human words. No one's published a single spike in a singing budgie. They're incredible. We got very curious about these animals and we started recording from people using both spiking and ECOG recordings, and thought, this seems to be an important missing link. And so let's think about what this bird might be doing. So I'm going to throw you a little bit of data from a lab meeting from a couple weeks ago from my postdoc, because I think it's really exciting. If anybody wants to come and play with budgies, please do. I think it's very early days, but I thought just as a whatever tiny cookie they might bring at the end of a meal, this is maybe that. Okay, so what we do with budgies, they're these animals. They are interacting in these reasonably large cages and arenas. We can't house them by themselves because they're enormously social animals, kind of like Dee Dee's marmosets. And we can put these piezo microphones on each one of the budgies. And the point of those is they do a very good job of trying to pick out who's contributing what to a very complicated soundscape. So this is, unlike the zebrafinch, we have this very neat and tidy motif that they've taken most of their juvenile months to learn, and many hundreds of thousands of trials. These guys can sing in an ad hoc manner, very creative, one-shot learning, all of these things. And so there's this very complex soundscape, who's contributing what. These microphones do a great job of saying, okay, this bird contributes that, this bird contributes that, and on and on. And so we can do recordings in these guys. This happens to be when the partner bird is singing in blue, and when the bird we're recording is singing in green. This is in the equivalent area of our QR code area in the zebrafinch. So this is called AAC. It's a very different brain, actually, but synaptically it's 2 levels up for muscle. We can see that it's really bursting during the time that the recorded bird is singing, as opposed to any kind of sensory input driving that activity. We can say, if we look at the amount of burst during singing versus not singing, it's much, much more. And this is across all of those neurons. And there's an enormous amount of structure there, right, in this part of the brain. So if we look across these 3 trials, and these animals are making a contact call. So this thing looks like this. When I try and read some of the languages I see in buildings, maybe it looks like this. I don't know. This is an English speaker's perspective, but you can see that if I overlay these, the parts that are gray are similar across trials. They're extraordinarily similar. But they're typically not making these contact calls. They're making what is referred to as a warble, and the warble can be really this creative, open-ended exploration of a space. So this is just a few seconds of warble. You can see they're moving all over the place vocally. This is just the animal that we're recording here that's got a piezo mic. And across 81 neurons, we see the structure here, right? As the animal is making similar sounds, there seem to be similar structure, and if we kind of informally package cells together that seem to be doing similar things, you can see that, for example, this swoop up is now the swoop down here of this ensemble, and so on and so forth. Similar sounds lead to similar activity in the network. And so if we look at just a stretch of sound and a stretch of population activity, you can see how different that is. Right? So this is in the zebrafinch, and you see this kind of series of bursts that in some sense is a representation of time. We can decode this and know where we are in the song, but there's no clear modulation based on what the animal is actually singing. If we look here, you can see the song in the spike tree, right? You can see these kinds of moments here that seem to resemble that. And so we can formalize this by just looking at spectral correlation, just looking at the sonogram here. And there are moments when these are quite self-similar, like this. You'll see that with the block of color of yellow in the spectral correlation. Moments of similarity. And this also happens in the zebrafinch, very similar kind of squawky song. If we look in neural space at similarity, we see that sonogram is really beautifully reflected here. And in the zebrafinch, things are evolving with really no history. Right? They kind of are doing what they're doing in this encrypted way. So the correlation between the sound and the neural in the budgie is extremely high, and the sound in the neural in the zebrafinch is basically a chance. Okay, so just to wrap this up, we have could try and look for just very low-level sound features. I talked to over Chernikovsky. He said just look at fundamental frequency. So if you could follow this, you can see the white line. We do a very good job of estimating fundamental frequency for this specific contact call localization. And we take all of the moments where this bird is singing, we break them into little pieces, 20 millisecond pieces, and we have 81-dimensional space. It's something neurons we've recorded from here. And we can just plot the first and second PC like that. And see, is there any relationship between network activity and frequency? And in fact, there is. Right? There's a beautiful pitch map at a network level that really shows the first principal component here, at least in this area of the brain. We would never see this kind of thing in the songbird. But some of these dots are gray. We can't really estimate them. And that's because some vocalizations are really noisy, right? Really high entropy vocalizations. Those are almost like white noise with this guy here on the left, minus 1.9, versus this lower entropy vocalization that we do well at establishing fundamental frequency. So if we just plot entropy, we see that there seems to be a population representing tone and frequency, and also the second principal component of a whammy bar, if you like. So just something about the quality of that tone, entropy in this case. And so when we think about how this is coding, there are some real similarities to human speech that we don't see in the songbird. So this is 2 synapses up, as we find here in the premotor cortex, and what Eddie Chang has done here, or Chris Beauchard, is use electrocorticography to try and characterize this part of the human cortex. So he sees articulatory maps of lips, jaw, tongue, larynx. And we see something here that looks like a similar kinematic map in the budgie. And so this is something that we think of it almost like a song keyboard. It exists here. It doesn't exist in the songbird. And we're very excited to now look one level up from this to see who's playing that keyboard and what gives rise to things like vocal flexibility, one-shot learning, and interactions. I'd like to thank the lab. This is the lab. Thank you. I can't believe you all stayed. This is see what you're going to think. Yeah.
So, regarding your thalamically neurons, I'm just wondering if you have looked very close at them with respect to when they fire relative to a syllable and the whole song. So is there some sort of a hierarchy there? Like, some that only fire at start and end of syllables, some that, like Dilap was asking, would sort of mark like the entire song?
Yeah. So let me just say a few things about that. So there are different types of initiation jaws that have to happen, and I've described one of them, which is syllables within this complex vocalization. But the bird is often, if you see a bird sitting there, you'll hear a lot of chirping, this contact call. It's a different vocalization. It's just kind of like, hey, where are you? Where are you? Where are you? Where are you? It's a very short kind of thing. Those thalamorecipient cells are at the beginning of those short vocalizations as well. There are another population of cells that are active when this animal is clearing its throat, and they call them introductory note. This animal is getting ready. You hear the engine going. There's another population that is actually initiating that vocalization as well. And so it seems that it's just a general dynamical initiator across different vocal behaviors in this animal. Those vocal behaviors are amazing, but somewhat limited. So we'd love to know how this works in a Bengalese bird, where order is more important, and maybe we can get to things like hierarchy at that point. But it's a great question. Thank you. Yes?
Sorry, a little question about learning. So, as far as I understand, these are adult birds, but do they need to learn the song, how to do vocalization? And if they do, how does this relationship change following learning?
Right. So these have all learned. We get them, having already learned from their fathers from Oregon. So we get them in a different state. They go on a plane and we pick them up here as adults. How this works in learning is something that I'm very excited to study. We don't know. And there are the ideas of existing tendencies within this area to form sequences that then are co-opted by behavior. If there is some kind of already latent synaptic connectivity that's going to make you do a pre-play or something, and then how does the bird actually use that when it comes time to making a behaviorally relevant sequence? We don't know, but we're going there, and so stay tuned. It's a great question. Thank you. Good. Thank you all.